In order to identify our unknown bacteria, we need to do some tests. So the first one is the catalase test in order to identify our bacteria. So here is the plate with catalase positive and the other one is catalase negative. These are two different bacteria. So in order to do this experiment, we need to add the hydrogen peroxide to clean glass slide, and then after we are doing that, we need, with a toothpick, we need to grab some bacteria like this, and then mix it with hydrogen peroxide. So the positive reaction is producing some bubbles, and the negative reaction is when you didn't see any bubbles. And as you can see, we are not able to see any bubbles. We are going to do the same thing for the catalase positive. So I will grab some bacteria, mix it with, oh, you can see the bubbles very quick. This is the next test for identify our bacteria is the oxidase test. So in order to do that, we need to put a filter paper on a top of paper towel and then add a drop of oxidase reagent to the filter paper, like this. Okay? Again, use a toothpick to smear some cells on a filter paper where we have the reagent. This one is the oxidase negative, so we wanted to see that. So, okay. So, if you see the dark purple color less than 30 seconds, it means the positive reaction. But here, we are not able to see the dark purple. Okay. So for oxidase test, here is the positive result, dark purple color, and here is the negative result. Test for identifying our unknown bacteria. So basically we have reagent A and reagent B and also zinc dust. And this is the three different positive and negative results. So the thing that we did is we added 5 drops of reagent A, 5 drops of reagent B to each of these tubes. So as you can see, this one, the color changed to red, but these two remain yellow. When the color has changed to red, it means the positive result for the reduction of nitrate to nitrite. But if it remain yellow, we need to add the zinc dust in order to see that if the reduction will continue to ammonia. So now we need to add a pinch of zinc dust to each tube. Okay. Okay. After adding the zinc dust, we need to wait a little bit. So if the negative result here is the red color. So as you can see, we can see the red color. So it means that the negative result, it does not reduce at all. But here you can see that the color hasn't changed. It remains yellow, which means the positive results and the ammonia and nitrogen are present. Lactose taste, basically for lactose fermentation. So the lactose can be fermented into organic acid and gases. Here is the positive result, here is the negative result. The positive result actually shows the acidic pH, and the red color, which is the negative result, is the neutral pH. Actually, the color hasn't changed in here, but it has changed to yellow for the positive. So here is the starch plate. Basically, we streak our unknown bacteria into 2 starch plates. in order to see the result, we need to add the iodine. If the amylase is present, then there should be no starch around, and we could see it after adding the iodine. So here is the iodine. Okay, so clearly we can see that there is no starch around where the bacteria can grow. Iron agar, which is the three tests in one experiment. Basically, if the hydrogen sulfate produced, we should see the black color like this. So this is for hydrogen sulfate production. You cannot see the hydrogen sulfate for the others. And yellow color shows the acid production. So in this one, the acid produced. And the cracks in the agar shows the gas positive. So as you can see, here is the cracks, which means the gas produced. And also we have and this one is the negative result. And also we have the O2 requirement, which could be aerobic, anaerobic, and microaerobic. So when the bacteria grows on the surface, which is the aerobic, if it grows like this, which is you can see that the bacteria grows all over the tubes and also on the surface. It means that the facultative, aerobic, or aerotolerance. Okay, and then you can see that the bacteria grows all over the tube and also on the surface, which means that facultative, aerobe, or aerotolerant, anaerobic. So this is another test for the enzyme that degrades gelatin. So here you can see that the negative result, and here is the positive. So the difference is when we put it on ice for 30 minutes, if it's become solid like this, which it means negative. But if it's still liquid, it means the positive result. Here is the test in order to see that if bacteria are able to hydrolyze urea to ammonia and CO2. So as you can see, if it's changed to hot pink, it means that the positive results and the ammonia is produced. So the pH will increase and you can see the hot pink. And this is the negative result, which we won't see any changes in the color. The test for the exoenzyme that coagulate plasma. So here is the positive and here is the negative result. So the positive result, which is solid, means the plasma is coagulated. But the negative result, which is liquid, it means the negative result. So here is the test for the enzyme that hydrolyze DNA. So as you can see, when DNA is hydrolyzed, the dye can no longer bind to DNA. So you can see the hollow around the bacteria. But here is the negative result, which you can see nothing actually.